Alright, so we're going to be talking about the cell membrane and what it does. So first, a little bit of history. Even though cells, we know about them from years ago, the cell membrane we actually did not learn about until more recently. So the reason why is because it was limited by what microscopes we could use. So early in the 20th century, the early 1900s, we did know that the cell membrane had phospholipids and cholesterol and proteins, but we didn't know how it all fit together. This is an early electron micrograph of two cell membranes. You have one cell on top and another cell on the bottom, and two phospholipid bilayers. So this is basically what they could see. So they knew about the phospholipids. They did not know how cholesterol fit into things, and they did not know how the proteins fit into things. What turned things around was a technique called freeze fracture. So freeze fracture was a technique that was developed that allowed them to basically cool the membrane down, really cold, and then they could actually pull it apart and see the two separate layers. And that led them to what we call the fluid mosaic model. The fluid mosaic model, if you're not familiar with what a mosaic is like a picture made out of tiles, where there's like cement and there's tiles stuck in it. I always think of when I go to Disney World, there's a mosaic of like Cinderella. When you walk through the castle, you can see it on the wall. But you've seen mosaics before. It's basically pieces of tiles. So the fluid mosaic model, the tiles would be like the proteins stuck in the membrane. And then the fluid part would sort of be like the cement. So that's sort of how the phospholipid bilayer is a mosaic. All right. So from the previous chapter, we already know that it's a phospholipid bilayer. The hydrophilic heads, that's your phosphates. They point outwards. And then the tails point inwards. They face each other. The other thing is, the phospholipids are unsaturated. What do unsaturated fatty acids have in them? They have kinks that are caused by the double bonds, exactly. So that's why the lipids in the phospholipid bilayer are usually drawn like this with little bends to represent the unsaturated fatty acids. And that keeps the membrane fluid. We usually think of the cell membrane as a solid. You probably think of it like a water balloon that is your cell and that the balloon part is the membrane. But technically the membrane is fluid. Things could not diffuse through it very easily if it was solid. So it's technically a loose, fluid-like membrane with the phospholipids moving around. The other thing is the proteins we now know are stuck in the membrane. So some of the proteins are on the outside. They're called extrinsic, like external. And then there's some that are called transmembrane proteins, which actually go all the way through the membrane. So they sort of stick out on both ends. These would be like the proteins, for example, that are carrying things through the cell, in and out of the cell. One final thing that the cell membrane has, in animals only, is cholesterol. Common question I usually ask on the test. It'll be something about what kind of membranes have cholesterol. Make sure you know that it's animal membranes that have cholesterol specifically. So when they brag on, a bag of pretzels that it's a cholesterol-free food, it's not like they magically took the cholesterol out of it. If it's made from wheat and oil and salt and flour and whatnot, those things don't have cholesterol in them anyway. Cholesterol is only in animal cells. But this is something that, in addition to the unsaturated fatty acids, will help keep the membrane liquidy. I was going to say milk. Oh yeah, that's an animal product, yeah. So there are some, I looked it up, there are some things called plant sterols that are, I guess, somewhat similar to cholesterol in their structure, but apparently plant sterols will actually lower your cholesterol levels. They're actually good for you. There are no plants that actually have cholesterol in them. This is a couple of diagrams of different membrane portions. So this is a membrane protein. This is just a reminder that although they draw membrane proteins usually as just like purple ovals and tunnels and things, in reality, remember, a protein really is a long long chain of amino acids that's folded up into a specific structure. Here you see the a section that specifically shows cholesterol. So I don't know if you remember this from biochemistry, but cholesterol looks like 4 fused rings. All steroids do. So these yellow little sets of 4 fused rings here, that's cholesterol in the membrane. And this last diagram is just a reminder of what a phospholipid looks like. In the biochemistry chapter, you learned what it looked like based on its chemical structure, like with the glycerol and the 2 fatty acid chains and then the phosphate. But in this chapter, we just represent it as the ball is the phosphate and then the 2 tails are the fatty acids. And then this is kind of a complex 3-dimensional one. You saw a picture like this before when we talked about the extracellular matrix. So all these purple things are representations of the proteins in the membrane. Some of them are transmembrane. They go all the way through. But like you'll notice here, they have a picture of a peripheral protein. Oops, trying to stretch it so you can see it. It's not stretching. Nah, it doesn't want to stretch. That's okay. This picture shows how unsaturated fats, because they're unsaturated, you see how there's spaces. This is why the membrane is fluid-like. So small things can diffuse through the membrane because there's actually gaps caused by these phospholipids. Whereas if they were saturated, it would be really tightly packed and stuff wouldn't be able to get through. Like oxygen that your cells need all the time would not be able to pass through the membrane if it was very tightly packed. And then this is showing how in animal cells you also have cholesterol in there. So cholesterol, even when it gets really cold and lipids might start to solidify, cholesterol can be sort of packed in there and help keep it also fluid. Another thing is that the lipids can move around. So again, we usually think of the membrane as being some kind of a solid, like a sac around the cell. But that's not really what it's like. So when they say in this statement here that the phospholipids can move laterally, what they mean is, if I was to draw like a little 3-dimensional section of membrane here, so let's say this is sort of a 3D section of membrane. If I was to code one of these phospholipids, let's say this one, and I colored it orange, and I was to watch it, it wouldn't necessarily stay in this place. Like the phospholipids can literally move around. So it could end up over here, or it could end up back here, or over here. What would not happen to this phospholipid is it would not flip over and end up on the other side that's facing the inside of the cell. And the reason that wouldn't happen is because since phosphate is hydrophilic, there's no way this phosphate's going to pass through this whole hydrophobic section here and then end up on the other side. So phospholipids facing the outside, even though they do move around laterally, side to side and around on the same side, they don't flip and end up on the other side. That's all that's trying to say. The other thing you should know is that the outside and the inside of the membrane are not the same as each other, which makes sense because there's different jobs. For example, on the inside, you might be hooking to the cytoskeleton to hold the cell shape. On the outside, you might need, for example, ID markers or receptors to pick up information from cells around them. So what's going on the outside of a cell is not the same as inside, so the proteins and the membrane structure don't necessarily look the same on the outside as the inside. All right, any questions so far? Okay. So, again, even within different organisms, you know certain foods are higher in cholesterol than others. One of the reasons why certain organisms may be higher in cholesterol may have to do with where they live. Remember, cholesterol helps keep the membrane fluid at low temperatures. So it's possible that if you were to eat fish that naturally live in a very cold environment, they may be higher in cholesterol than other fish that naturally live in tropical environments. You may also find plants that live in colder environments have more unsaturated fatty acids, like polyunsaturated fatty acids, because that's going to keep them more fluid when the temperatures drop. So this is why not all organisms necessarily have the same amount of cholesterol or the same amount of fatty acids, because their membrane structures vary depending on where they live. All right, proteins. So in the membrane, the proteins, as I mentioned, they fold up specifically so that the hydrophilic parts that, like water, sort of stick out with the phospholipid, with the phosphate part, and the hydrophobic portions sort of end up on the inside. They fold into very unique special shapes that give them their jobs. So we're going to go through 5 specific jobs that proteins in the membrane perform. All right, so first job of proteins in the membrane, transport. In other words, proteins in the membrane carry things in and out of the cell. Sometimes transport proteins require energy depending on what they're carrying. So a lot of times these will be drawn as literally like a little tunnel in a cell. So it'll sort of show it like this and show that some molecule can move through it. Or it could be shown, you'll see one on the next slide, where the protein looks like it carries things, like it changes shape. And a lot of times they'll put like a little arrow and write ATP there, showing that energy is needed for that protein to function. But those are proteins that are carrying certain things in and out. Most things cannot pass through the phospholipid bilayer. So they are relying specifically on the proteins of the membrane to carry them in and out. Most of the things the cell needs are not going to be able to pass through the lipid part. All right, an example that the textbook always uses of a disorder that's related to a faulty protein is cystic fibrosis. You will not be tested on specifically knowing cystic fibrosis, but it's sort of the textbook example. Cystic fibrosis has to do with a protein that specifically carries chloride ions. Normally, the way that protein folds up, it forms sort of a channel for chloride ions to freely pass out of the cell. People with cystic fibrosis, that particular protein has the wrong shape. It's got one amino acid is wrong, so the R group is wrong. The whole thing folds up wrong. I have a little diagram of it here. So rather than being folded in such a way that it forms a channel for chloride ions, it folds in such a way that it sort of blocks their transport. When it does that, a chain reaction happens, a series of events, but the outcome ends up that you end up with like this mucus buildup. People with cystic fibrosis have lots of mucus that lines their respiratory tract. They have a reduced respiratory function. It can be like having pneumonia all the time. They usually have to take medications for it. I know most people that have it have these special machines that they use in the morning and at night that literally shake their chest. They sit in this machine and it shakes the mucus loose so they can cough it up. Like it's massive amounts of mucus. And also in the digestive tract, same thing. It's an extreme example of a disorder related to proteins in the membrane, but it's just one example of why they're so important. So transport proteins carry things through. Second function, enzymes. Enzymes just speed up reactions. It's actually what our next chapter is going to be about. So some reactions happen right there on the surface of the membrane, and those would be catalyzed or sped up by enzymes. The third is support fibers. Those would be all of the junctions, like we talked about in the last chapter, gap junctions, desmosomes, tight junctions, and also the proteins that anchor to the cytoskeleton on the inside or the extracellular matrix on the outside. So they give the cell its shape and hold it in place. Receptors are proteins that pick up signals. An analogy of a receptor would sort of be like my doorbell. If somebody rings my doorbell, they don't actually enter my house to do that. They push a button on the outside. They never actually come in my door. But when they push that button on the outside, which is sort of like what a receptor does, a receptor protein, this would be like a hormone. And this is sort of like the hormone ringing the doorbell. When that protein gets stimulated, some kind of response or reaction happens. Just like when somebody rings my doorbell, a bell goes off inside the house, my dogs start barking, I go check to see who it is. A whole bunch of stuff happens in response, even though the person ringing the doorbell never actually came into the house. Same thing here. A hormone can bind to a receptor, and then your cell will carry out a reaction. Insulin, for example, binds to receptors. When it binds to the receptor, the inside of the cell